Welcome to this seminar on quality control in a laboratory context with me, Gary Moser, Quality Manager at Latis Scientific. When talking about quality control or QC, there is sometimes confusion between this term and quality assurance or QA. To clarify, QA refers to the policies, procedures and other measures, for example audits, that assure the maintenance of satisfactory quality, whereas QC denotes the practical checks carried out in order that sound analytical data is produced. Quality control is applied in many industries, for example in manufacturing, in order to produce products of a specified quality. In the context of a laboratory, therefore, QC is essential to producing analytical results that are fit for purpose, meeting customer expectations in terms of their veracity and accuracy, reliable so that sound decisions could be made on them, consistent so that if determined on different batches, results should be comparable, provided that the determinants are stable enough, and to the expectations of the ISO 17025 standard to which Latis Scientific is externally accredited. ISO 17025 has a large number of requirements. This slide will give you an idea of the QA and QC requirements of the ISO 17025 standard, and so what is expected by UCAS. Note that this is not an exhaustive list of ISO 17025 requirements. Later in this session we are going to consider external quality control, but first we will look at the internal quality control of laboratory processes. Quality control originated for industrial applications and is the use of statistical tools to monitor process performance. A technique often referred to as statistical process control or SPC is widely used in manufacturing and was proposed initially in the 1920s by Walter A. Shewhart, who introduced the concept of the process control chart. The chart type that bears his name, and that remains the most common type in use to this day, shows graphically if a process is performing within set limits and can also indicate anomalies, step changes, and any developing trends in performance. Corrective or preventative actions may then be applied. This is a typical Shewhart chart, and would look much the same whether it was monitoring the performance of a laboratory method or that of an industrial process. The individual points plotted are the QC results for each batch. We'll come back to this and consider how it's used to control a process in more detail in a short while. For a laboratory process specifically, unlike for manufactured goods, we can't carry out QC measurements against product specifications, as our process outputs are analytical results and the samples these relate to, by their very nature, unknowns. Therefore, AQC samples of known composition must be added to all batches of analysis. These should be matrix matched to be as similar as possible to real customer samples, and must have the compounds of interest present at known levels, preferably at concentrations that reflect levels in real customer samples. They are prepared and processed with each batch of samples, and the results plotted on a control chart. The control limits for the chart are statistically derived from previous real data. This laboratory application of SPC, usually referred to as Analytical Quality Control or AQC, constitutes our internal quality control for test methods. So, the AQC samples we employ are samples of known type, composition and determinant concentration included with each analytical batch to demonstrate that the method is performing satisfactorily. Having the determinants intrinsic to the sample rather than added or spiked onto a sample is always preferred. Such samples can be certified reference materials from external suppliers, although the availability of these may be limited and they are often too expensive to use in routine QC. Or in-house reference materials prepared from already characterized samples if the matrix and determinants are sufficiently stable. Alternatively, certified standard solutions are accurately spiked onto a typical sample matrix, preferably blank with respect to the determinants of interest prior to spiking. This technique is often used if determinants of volatile are unstable. For microbiological methods, AQC samples must also be prepared fresh from proprietary certified reference materials that are cultures of the organisms of interest. To implement quality control for a method, we need to apply some statistics to a data set of AQC sample replicates. Our primary statistical model for AQC is the normal or Gaussian distribution. If you've studied statistics, you will have encountered this before. Put simply, more replicate results are likely to fall near to the mean average of a data set, while moving away from this, fewer results will be expected. The other important statistical concept for QC is the standard deviation, referred to as the SD, sigma, or often just S. This value indicates the degree of variation in the data set to either side of the mean. We can use the standard deviation to set up control limits on our Shewhart chart, since, for a process that follows a normal distribution, the following is always true. Approximately 68% of results will fall within plus or minus 1 sigma. 95% of results will fall within plus or minus 2 sigma. And 99% of results will fall within plus or minus 3 sigma, provided that the process in place is robust and is followed consistently. Represented graphically, it would look like this. So, 99% of results would be expected to fall within the 3 sigma limits, 95% of results fall within the 2 sigma limits, and 68% of results fall within 1 sigma. Now, if we turn our distribution through 90 degrees, and we think back to our Shewhart chart from earlier on, then things start to look familiar. Basically, if we remove the lines drawn at plus or minus 1 sigma, we have a Shewhart chart. Just to clarify, the abbreviations used against the lines on this chart are UAL, the upper action limit, and LAL, the lower action limit, at plus and minus 3s respectively, and UWL, the upper warning limit, and LWL, the lower warning limit, at plus and minus 2s. Note here that some sources refer to action limits as control limits and abbreviate them accordingly as UCL and LCL. The basic rules used to control any process are shown here. These are applied as each data point is added. There are other rules that might be applied in addition to these, especially during periodic chart reviews. For example, the more advanced rules promoted by the prominent American statistician and advocate of the use of SPC in industry, James Westgard. But for this session, we will concentrate on those rules familiar to all users of such control charts. So, if we encounter one point outside of plus or minus 2s, then the next result is monitored, but on its own the first does not constitute a failure. However, if the second point also falls outside of plus or minus 2s, then a failure has occurred. A single point outside of the plus or minus 3s limits is an immediate failure. In the event of a failure, samples associated with the failing AQC are retested. All failures must be formally investigated and documented. An investigation is conducted by a specialist in the particular method or lab technique, a senior analyst or above. And following this, the quality team review its outcome and have the final sign-off of both the investigation and any proposed corrective actions. Shewhart charts need to be set up carefully, monitored and maintained in order to function properly. Initial chart limits are set from method validation data or from at least 20 early AQC results. More robust limits are set after 60 points, reviewed after 100 to 200 points, and then reviewed periodically thereafter. Striving towards zero defects is a laudable goal, but statistically a small percentage of failures are bound to occur, as we discussed earlier, such that 1 in 100 points is expected to fall outside of plus or minus 3 sigma limits, on average over time, and 5 in 100 points outside of plus or minus 2 sigma. Trends developing over time or any sudden step changes are also monitored on these charts, which would result in an investigation and corrective actions as for other failures. Internal AQC, while crucial to ensuring the quality of reported data, is however only one aspect of laboratory quality control. We will now consider external QC. This can be by means of inter-laboratory trials between partner organizations, often known as benchmarking or round-robin exercises. But the preferred means is proficiency testing, or PT, which is truly independent and carried out blind. In some contexts, such programs may be more commonly referred to as external quality assessment, or EQA. Latis Scientific participates in a number of PT schemes, of which these are some examples. All participants are sent identical samples by the scheme. The true results are not published in advance, and participants are anonymous on the scheme's reports, each lab identifying its own results by means of a code number. Performance in each test for each determinand is expressed as a z-score, and here you can see the calculation for this. A z-score of 0 means the results fall exactly on the assigned value, i.e. the true result as assigned by the scheme. Like with our Shewhart chart, if a result in one PT round falls outside of plus or minus 2z, then the next round is monitored for the same determinand, but no failure has occurred. However, if on 2 consecutive rounds this determinand score falls outside of plus or minus 2z, then this is a failure. And if a result outside of plus or minus 3z is produced, then this is an immediate failure. Failure investigations are conducted by a senior analyst and managed by the quality team as for AQC failures. Just as for internal QC, trends in PT scores can be monitored more easily if we plot them on a chart. This trend chart illustrates how a method that would initially have been considered out of control from a PT perspective with respect to one of its determinants might over several rounds be brought back into control as improvement actions are implemented. Using PT does have some limitations. If there are too few participants, then the resulting statistics are not robust. If different labs use alternative methods, data must be segregated by method or the statistics may be misleading as they are then comparing differing processes. If labs' results differ widely, Z-scores may appear acceptable even if the reported result is actually far from the assigned value. The PT sample may not be representative of real customer samples, as PT samples are not readily available for all sample types. This leads to some being merely standards in a suitable solvent or acid matrix. PT samples may require non-standard dilution or preparation, for example, for unstable determinants, which are generally transported as concentrated solutions. Volatile analytes in particular are nearly always spiked onto a sample matrix, so additional variability is unavoidably introduced. However, there are a number of very good reasons for using PT, and all good laboratories will have an extensive PT program, like ours here at Latis. PT provides a means of assessing a lab's performance compared to other labs that undertake similar testing. It is independent of the laboratory's own internal AQC, and so may detect problems with the internal AQC process. The ISO 17025 standard and UCAS advocate the use of PT. Indeed, UCAS assessors will always ask to see a lab's PT scores when they audit. If PT is not employed, then some other form of external QC is seen as prudent. For example, periodical tests using CRMs, which can in fact be previous PT rounds. And finally, good PT scores are a strong indicator of sound performance, as it is conducted blind and independent of the organisation. That concludes our seminar for this session. Thank you very much for your time today and for listening. If anyone has any questions on AQC and PT, I shall be very happy to answer them.